Welcome back to part 4 of chapter 17. In this video, we'll cover section 17.4 on pathogen recognition and phagocytosis. We're going to look at how leukocytes detect pathogens, leave the bloodstream, and destroy microbes. By the end of this video, you should be able to explain how leukocytes migrate from the peripheral blood into infected tissues, describe how leukocytes recognize pathogens, and walk through the full process of phagocytosis and how phagocytes kill pathogens. This first section on pathogen recognition and phagocytosis focuses on phagocytes, and these are innate immune cells that detect, ingest, and destroy pathogens. The major phagocyte types are neutrophils, macrophages, and dendritic cells. And we're going to review 4 key processes in this section: extravasation, pathogen recognition, phagocytosis, and microbial degradation. Extravasation, also called diapedesis, is the movement of leukocytes or white blood cells from the bloodstream into infected tissues. So down here I can see part of a blood vessel. Here is a white blood cell, and I can see it's leaving the blood vessel to enter the tissues. Here, that must be the infected site or the infection site. This is triggered by complement factor 5A and other pro-inflammatory cytokines that are released at the infection site. This leaving of the white blood cells can only occur in post-capillary venules. It does not happen in arteries or veins because their walls are too thick to cross. And if you read the book, you'll note that they actually say it happens in capillaries, but that's not quite accurate. Post-capillary venules is more accurate. Of the leukocytes, neutrophils will arrive first, followed by monocytes, which are going to differentiate into macrophages at the tissue site. The steps of extravasation are very structured. We have cytokines that increase the adhesion molecules that will be expressed on the inner lining of that capillary endothelium. Leukocytes, because of these molecules, will slow down and start rolling along the vessel wall. And where there are cell junctions, one endothelial cell meets the next cell, there will be adhesion molecules that cause very firm binding. So the white blood cells will be firmly attached. Then the leukocytes will perform transendothelial migration, where they squeeze in between those endothelial cells, you can see that over here, to enter the tissue. And once they are outside of the vessel and in that infection site, or moving towards that infection site, the leukocytes know where to go because they're following chemotactic gradients created by the C5a and other pro-inflammatory cytokines. So they follow them kind of like following breadcrumbs on the ground toward a higher concentration, and this migration ensures that the immune cells will reach the exact location where they're needed. In a second, I'll show you the figure that summarizes leukocyte migration, but before we look at that, I wanted to show you or summarize the process you're going to see. First, we have damaged cells and resident macrophages that are releasing those chemotactic cytokines. Complement activation will generate that C5a factor, and that's going to attract leukocytes to the damaged area. Leukocytes will exit the blood vessel, and then we'll have neutrophils, once they're at the tissue site, release chemicals and start phagocytosing microbes. Monocytes will later enter the tissue site and differentiate into macrophages to continue clearing the site. So this slide shows those major steps visually. Here we have leukocytes in the blood start detecting those chemical attractants. They're going to squeeze in between the endothelial cells, and once they get into the tissue, neutrophils will degranulate, release those toxic chemicals, and phagocytose the pathogens. So this is the key mechanism that gets immune cells from the blood to infected tissues quickly. If you're interested in seeing that in action, there is an animation that you can see at this link. I've taken a screenshot where you can see white blood cells binding to those adhesion molecules and squeezing in between the endothelial cells to reach the infection site. You can they also include a real microscopy video showing leukocyte rolling. So here is the blood vessel. We have a bunch of leukocytes here. Some of them you can see in the video will squeeze in between the endothelial cells, and there are a few leukocytes already at the infection site. This checkpoint asks, explain the role of adhesion molecules in the process of extravasation. So during extravasation or diapedesis, adhesion molecules on those endothelial cells and the leukocytes allow the white blood cells to leave the bloodstream and enter the infected tissues. So the role of adhesion molecules, we start with cytokines. Cytokines are released at the infection site, and they cause those endothelial cells lining the blood vessels to increase their expression of adhesion molecules on their surface. These molecules make leukocytes slow down, and they're going to start rolling along the vessel wall. Stronger adhesion molecules help leukocytes firmly attach, and they are going to stop at the junctions between endothelial cells. So adhesion molecules are going to allow the white blood cells to squeeze through the endothelium and enter the tissues. So bottom line is the adhesion molecules slow down the white blood cells, allowing them to stop and squeeze out so that they can exit the capillaries and reach those infection sites. Now let's look at pathogen recognition. This is how phagocytes recognize pathogens, and there are two main strategies. One is opsonin-dependent recognition. This is when pathogens are coded with opsonins, including antibodies or these complement factors or proteins like C1q, C3b, C4b, or lectins. The second strategy is opsonin-independent recognition, and that is when phagocytes can detect PAMPs. The PAMP stands for pathogen-associated molecular patterns. These are basically molecules that are found only on microbes and not normally found in our bodies. Going back to those opsonins, remember that opsonization is basically tagging pathogens for easier phagocytosis. So these opsonins can help coat the surface of our pathogen, making it easier for the phagocyte to recognize and bind to. For the opsonin-independent recognition, remember this is through PAMPs. PAMPs were pathogen-associated molecular patterns that are found on microbes, but not normally found in our body or on our cells. So these are microbial molecules that are shared across many different types of pathogens. So the major types of PAMPs I would like you to know include peptidoglycan. We saw that on our bacterial cell walls. Flagellin is also a PAMP. This is found on bacterial flagella. Lipopolysaccharides, we've seen that before on that outer membrane of gram-negative bacteria. We also have lipopeptides. These are expressed on most bacteria, where we can see them in their cell membranes or their cell walls, especially in gram-positive bacteria. And viral DNA or RNA, and those are nucleic acids that are found usually not methylated, as opposed to our molecules, which will have methyl groups or other associated proteins that we can tell apart from viral DNA versus RNA. So the detection of these pathogen-associated molecular patterns or PAMPs tells the immune system that a microbe is present. So how do phagocytes recognize those PAMPs? It turns out that phagocytes express pattern recognition receptors, or PRRs, including one of the most famous kinds called toll-like receptors, or TLRs, and that is what they use to detect those PAMPs. For these PRRs, these pattern recognition receptors, they can be found either on the plasma membrane of the phagocytes or inside the phagocyte's phagosome or other internal membranes within those cells. When these pattern recognition receptors bind to PAMPs, they send signals to their nucleus, and this will activate the genes responsible for phagocytosis, cellular proliferation to cause cell division of these phagocytes, cytokine release, interferon production, as well as intracellular killing. Once activated, these phagocytes will transition into a hyperactive state. And again, this is when those pattern recognition receptors are binding to the pathogen-associated molecular patterns. So there will be a greater rate of phagocytosis. The phagocytes will divide. They're going to make more and release more interferons and cytokines. Remember, interferons are used to prevent or released to prevent viral spread. There will be enhanced intracellular killing. So this is inside of the cell because some pathogens can survive and continue to divide within a cell. These PRRs can also respond to distress signals from damaged host cells, so during the damage of our own cells. This first checkpoint asks, name 4 pathogen-associated molecular patterns, or PAMPs. So we have peptidoglycan, found on bacterial cell walls, flagellin, found on bacterial flagella. We have that lipopolysaccharide, that LPS layer, found on gram-negative outer membranes, or they could be viral DNA or RNA, which are usually not methylated because our DNA is usually methylated. Or viruses sometimes, remember, have double-stranded RNA, which we don't usually see, or single-stranded DNA, which we don't see in our own cells. So these are conserved structures that are found in many microbes. For the second question, describe the process of phagocyte activation. So phagocytes become activated when their pattern recognition receptors, or PRRs, bind to pathogens, or they can also respond to signals from our damaged cells. So usually PRRs are going to bind to and detect PAMPs, and then this binding will activate intracellular signaling pathways. So the phagocyte will shift into that hyperactive state. They're going to increase their rate of phagocytosis. They're going to increase their secretion of cytokines. They'll be able to kill microbes more efficiently. And they're also going to eventually trigger the activation of cells we're going to talk about more in Chapter 18, like macrophages and dendritic cells, which are going to sweep up the pathogen and hold them out, present out those pathogens to alert our immune system that is more specific. So activated phagocytes overall are more effective at destroying pathogens. Now let's walk through the steps of phagocytosis. First, the phagocyte will attach to the pathogen using those pattern recognition receptors or by binding to opsonins. Pseudopods will be extended from the phagocyte around the pathogen. The picture in our book didn't really have a good one, so I took one over here. You can see those pseudopods extending around the pathogen. Engulfment will occur, so the pathogen is engulfed and surrounded by the phagocytic cell membrane. That is called a phagosome. So that structure is a phagosome. The internal compartment of the phagosome will become acidic, drops to pH of around 4 to 5. So this is known as acidification, or an early killing step. And then that phagosome is going to fuse with a lysosome. So lysosome, remember, these organelles contain digestive enzymes. When they fuse together, that is called a phagolysosome. So this process isolates the microbe and prepares it for destruction. We saw the formation of that phagolysosome. Let's now look at pathogen killing within the phagolysosome. And this involves multiple mechanisms. We saw acidification earlier. This is going to activate the enzymes within the lysosome. And the enzymes include lysozyme, phospholipase, proteases. All of these are capable of digesting the pathogen. There will also be a reaction called the respiratory burst within this phagolysosome. And what will happen is the phagocyte will produce many antibacterial reactive oxygen species, or ROS, such as superoxide anion or hydrogen peroxide. Many of these we've talked about before. That respiratory burst also includes reactive nitrogen species. Reactive nitrogen species including peroxynitrite, and all of these reactive oxygen species and reactive nitrogen species will combine to damage microbial membranes, damage their proteins, as well as DNA. So this combination of lysosomal enzymes, ROS and RNS, ensures that the pathogen is killed efficiently. All right, so we saw that phagocytes engulf the pathogen, forms that phagosome, fuses with a lysosome to form a phagolysosome, destroys the microbe, and then they're going to expel the pathogen components through exocytosis. At the same time, neutrophils can also release defensins externally to kill microbes in the tissues. We also have macrophages and dendritic cells that are going to keep some of these antigen fragments, these microbial fragments, for antigen presentation, and that's going to help link the innate and adaptive immune systems we're going to talk more about in Chapter 18. And if you're interested, our book provides a video showing a white blood cell chasing bacteria around. It's actually a neutrophil chasing Staphylococcus aureus. It is actually quite satisfying when he finally gets it. I don't know why I said he, it, the neutrophil. So take a look. The video is pretty short, about 30 seconds long. This checkpoint asks, what is the difference between a phagosome and a lysosome? So a phagosome is a membrane-bound vesicle that's formed after a phagocyte engulfs a pathogen. So internally or initially it contains that ingested microbe. And then we saw acidification where the pH drops to about 4 to 5. For a lysosome, that's a cytoplasmic organelle that contains digestive enzymes like lysozyme, phospholipases, or proteases. Inside lysosomes, we also have reactive oxygen species generating enzymes, so enzymes that have the ability to produce those ROS. And when the lysosome and phagosome fuse together, we get that phagolysosome. This is where the microbe is killed and digested. But some pathogens can actually survive phagocytosis. They can evade the killing that usually happens within phagolysosomes. And one example is Leishmania. Down here on the bottom left, the book shows a picture of cutaneous leishmaniasis. And instead of dying, these organisms, these are protozoa, can inhibit that respiratory burst and inhibit lysosomal enzymes. So within a phagocyte, this one happens to be a macrophage, they don't get killed or destroyed within those phagolysosomes. Instead, we can see that this protozoan has continued to divide within the macrophage. And I can also see here that these are nucleated, so they're not bacteria. These are eukaryotes, protozoa. So this highlights why some diseases can be very difficult to eliminate, especially when they cannot be killed within our phagocytes. So some key takeaways from this section, we saw that leukocytes or white blood cells reach infection sites through extravasation and diapedesis. The pattern recognition receptors found on phagocytes detected PAMPs, pathogen-associated molecular patterns, alerting the phagocytes that pathogens are present. Phagocytosis leads to microbe destruction through acidification, the enzymes found in those lysosomes, reactive oxygen species, and reactive nitrogen species. And macrophages and dendritic cells, we're going to see that they also function as antigen-presenting cells, and we'll see them more in the next chapter on adaptive immunity. All right, let's recap section 17.4. The first learning objective asks, explain how leukocytes migrate from peripheral blood into infected tissues. So this process was called extravasation or diapedesis. We saw that there's some kind of infection that occurs and cytokines and that complement factor C5a will be released. That causes endothelial cells to express adhesion molecules. We'll see leukocyte rolling, where the white blood cells slow down. They're going to roll along the capillary walls through weak adhesion. And then there will be strong adhesion where the leukocytes bind tightly to the endothelium, where transendothelial migration or diapedesis will happen when they're squeezing in between endothelial cells to enter the tissues. Then there will be chemotaxis to the infection site, where leukocytes are going to follow the chemical gradients of those C5a and cytokine molecules, and this only happens in post-capillary venules where the cell layers are thin enough to allow this migration. For the second learning objective, they ask, explain the mechanisms by which leukocytes recognize pathogens. So leukocytes use two main recognition strategies. We saw opsonin recognition or opsonin-dependent recognition, where, remember, pathogens are coated with opsonins, which make phagocytosis easier. And example of opsonins include the complement proteins like C3b, C4b, or C1q. They can also be coated with antibodies or lectins. And phagocytes have receptors that specifically bind to these opsonins. The second mechanism was the opsonin-independent recognition, and that is through the PAMP recognition. So phagocytes have those PRRs, those pattern recognition receptors, that detect the PAMPs, and PAMPs were pathogen-associated molecular patterns. And these included things like peptidoglycan, that lipopolysaccharide, flagellin, viral nucleic acids, or bacterial lipopeptides. And PRR activation, so those pattern recognition receptors, when they're activated, they caused an increase in phagocytosis, greater cytokine release, basically that hyperactive state of phagocytes. The third learning objective asks, explain the process of phagocytosis and the mechanisms by which phagocytes destroy and degrade pathogens. So if I look at the steps of phagocytosis, I have 1, attachment. Right, phagocyte binds to the pathogen using those PRRs or opsonin receptors. 2 was engulfment. The phagocyte extends those pseudopods around the pathogen. 3 was phagosome formation. The pathogen's fully internalized now, and it's in that phagosome, that membrane-bound vesicle. The pH drops to 4 to 5, starting that microbial killing. The fourth step was that phagolysosome formation, right, when the phagosome fuses with the lysosome to create that phagolysosome. So within that step, we saw some pathogen destruction through acidification, where the low pH can denature microbial proteins. There were also those lysosomal enzymes that were activated, like lysozyme or phospholipase or proteases, that can break down the cell walls, membranes, and proteins of microbes. We saw those reactive oxygen species and reactive nitrogen species can also further kill the pathogen. And there are also phagocytes can release defensins, which can kill pathogens outside of the cell as well. Then we finally had waste removal. So the digested material can be recycled, and the debris can also be expelled by exocytosis. So overall, we can see that for all 3 of these learning objectives, white blood cells leave the blood through extravasation. They can recognize pathogens through those opsonins or those PRRs that detect those PAMPs. For phagocytosis, we formed that phagosome, that phagolysosome, and there was destruction of the pathogen using acid, enzymes, reactive oxygen species, reactive nitrogen species. And that takes us to the end of our 4th video. In the next video, we're going to move on to section 17.5 on inflammation and fever.